from power of stem cells. So the first question I have to you guys is, what are stem cells? Any idea?
They come from bone marrow.
They come from bone marrow. Very good. Anything else?
Embryonic.
Do we have something embryonic stem cells? Is that Michael back there? No, Matt, are you raising your hand?
Yeah, cells with the
Wait, no, Matt. Not the right. You're at the reading part. So let me continue here. So stem cells have two things that kind of sets them apart from all the other ones. First of all, they have the potential to develop into any kind of cell. And the second one is they can renew themselves through cell division. So if we have a little diagram, what you see right here is, here's my stem cell, and the stem cell can either mature into whatever cell it wants to be, or it can just keep making its own type of completely kind of like bland cell. So fertilized egg is the ultimate stem cell. Any idea why? Everybody knows what a fertilized egg is? Any idea why that one is the ultimate stem cell?
It's made into all the different types of cells in the body.
Very good. It can go into anything. So, here we go. So, what's necessary to make a human being? What do we need?
fertilized egg.
Okay, we need a fertilized egg. So, in other words, we need two things. In order to fertilize the egg, what is the part that does the fertilizing? There you go. What's the other part? All right. So, we need a sperm and an egg. What does sperm and egg bring to the new human being? Anything? They bring DNA to it. Very good. So they bring a lot of DNA. 50% will come from the mom, 50% will come from the dad. What's the function of the DNA? Why is it so important to bring all that DNA to the cell? What about the genes? What do you need them for? Help her out. Any idea why we need all those genes?
Directing the growth and structure of these
Very
cells.
good. Yes, we want to direct something. We need to have a plan. So in other words, the reason why we need the DNA is it's a blueprint for the new human being, so the human being knows what it's supposed to be looking like. All right. So, question is, how does this fertilized egg look different? By the way, we call a fertilized egg a zygote. How does it look different from the full-grown human being? So here's your zygote. Totally unspecialized, non-committed cell, and then all of a sudden you turn into this human being which is made out of many different specialized cells. So the question is,
Would Jake Hollenbeck please come to the library?
how do you get from one completely unspecialized cell to an organism of about 50 trillion cells? So all of us in here have about 50 trillion cells, and those 50 trillion cells share about 200 different jobs. The question is, how do you get from that to the other one? Any idea? But if we only divide, then all we're going to get is more zygote looking like things. Nothing will look different. Signals are good. Let's go on that one. All right, so question is, how does the DNA of the zygote of a kidney cell look different from the DNA of a nerve cell, from a muscle cell? We have all these tissues in our body, 200 different types of things. How does the DNA differ?
DNA is the same.
Sam?
The DNA is the same.
Very good. So what's different then?
Which genes are activated?
Excellent. All right, we've talked about this before. Okay, so no difference. All those cells have the exact same type of DNA, but what is different is, here we go, what part of the DNA actually is made into protein. So if it's not the type of DNA that is present in each of those cells, that DNA is completely identical. What then is different? And what's different is kidney cells have a different part of the DNA made into protein. Nerve cells have a different part of the DNA made into protein. And if you want to look at something like this, like say this thing right here, which is, any idea what that's supposed to represent? Your whole DNA. Let's just make this up. Let's just say this is the entire DNA we have in any kind of given cell. The idea is, if I take this and make this into protein, it's going to be developing into a kidney cell. If I take this part of the DNA, it's going to be developing into a nerve cell. So in other words, different parts of DNA are used to make different types of cells. But they all have this DNA in there. It's just I'm not reading the entire strand. All right, so a little bit about human development. Go back to our fertilized egg. And here's a little sperm trying to get to the egg. And fertilization happens about here. And then over the course of about 4 to 5 days, we get all the way down to this part right there. So we're starting out as simply an egg with a sperm inside, and then it divides and it divides and divides, and eventually what we look like is a hollow ball. I'm going to try to find a ball in Mr. Westby's room, and we can only come up with this thing, so it's got to be doing for our model. So we all look like this at some point, but a heck of a lot smaller. This is about after like 6-7 days, that's what we look like. All right, so same thing here again. Here's my fertilized egg, and then we divide and divide. Here's my hollow ball. At this point, all the cells in that hollow ball are pretty much doing the same thing. They all look the same. Nothing has been changed yet. And then what happens is the following, and this is the part where we needed a model, but it didn't really work. Think about you take this ball and you're pushing the ball in. And as you're pushing the ball in, it was a little easier with the balloon. What you're doing is you're all of a sudden forming this thing right here. Can you guys see this? Like I'm pushing the ball in, and it turns into this. Eventually it'll turn to something called gastrula. And at this point, that's the first time where we have 3 different types of cells. And those 3 different types of cells, you see a little bit better here. Here's my cell balls. Then we start to kind of like push stuff in, and all of a sudden we see 3 layers. We see an outside layer, an inside layer, and then we see this middle layer right here. So we have 3 different types of cells at this point. What have the cells done to this point? Any idea? What do we call that? Those are in AP Bio.
Differentiated?
I'm sorry?
Differentiated.
Absolutely. These guys have differentiated at this point. So at this point, I have 3 different types of cells, and I call this whole thing now a gastrula. So the gastrula has 3 different layers. It's a little bit harder to see right here, but there's the outside layer. I'm starting to shake, so I have to go over here. So outside layer, inside layer, and then this middle layer right here. And this is where we start to differentiate even more. What's now starting to happen is
Taylor Garlock and Brittany Sykes, please report to the main office. Taylor Garlock and Brittany Sykes, please report to the main office.
those different layers from that ball of cells that we have, they give rise to different types of body tissues. And I don't know whether you guys can read this. I'm having a hard time seeing this. Can everybody see what it says? Nobody can see it, but we can recognize a couple of cells. Mr. Hamele, we cannot make this a little bit easier.
Yeah.
And you know what, maybe we don't need to. Let's just,
We're good.
all we can see is we have, what we start to do is we start to differentiate. So this one is going to be, this is the ectoderm, the outside, this is the middle part, this is the outer part, and they start to go into the different part that make up the human being. So at this point what we have done is we're starting to go from 3 types of cells into more and more specialized, more differentiated types of cells. So at this point cells are differentiating into tissue-specific cells and take on specific jobs. Almost like when you guys are first born, you're basically a clean slate. And then you go to school, you get a little bit more specialized, and you go to high school, you're a little bit more specialized, you go to university, a little bit more specialized, and finally by the time that you've gone to your first job training, you're super specialized and you work in that job. Similar to this right there. From the beginning on till you're very specialized in your job, what have you gotten every single day? Instructions, signals, same thing with a cell. So the question is, how does a cell know to do that? They get this, specific signals that allow them to turn certain proteins, certain genes into proteins. So here's the same picture again. Here's my fertilized egg. Here's my gastrula. And what we have right here, those 3 different types of cells. And these 3 different types of cells become a little bit more special and give me 3 different more types of cells. And these 3 different types of cells, then this one develops into lung, pancreas, heart, blood, and so forth. So we start to specialize more and more and more. And the types of cells that are doing this are what we call our stem cells. Go back to this one. So right here, as I told you guys, remember I started out saying that the fertilized egg is a completely uncommitted kind of cell. We call it a totipotent cell. Totipotent means it can do anything. It can do any kind of job. Then it becomes a little bit more committed. It loses a little bit of its potency. We call it now pluripotent. All the way down, it has completely specialized and it's unipotent. So the analogy would be here is, as a baby, what are you? What potency do you have? Totipotent. And once you're working a job, like all the teachers here, unipotent, all the way specialized. And from here to here, how do we get there? We get signals. We get little things that tell us do this, do that. All right, so what makes a cell to commit to become something special? These little green things, remember all the apibiotic people, where they remember? We had them once? Little signals. So, two sources of information basically tell the cell which genes to express at any given point in their development. And the two types of signals are signals that are actually coming either from inside the cell or they come from the outside the cell. An outside the cell kind of signal could be growing next to another cell. That's a signal. If I bump into something, I'm giving you a signal. Or I might have something coming in from my inside. That's another type of signal that makes me do something different. Same thing over here. But the important part is, no matter what the type of signal is, what the origin of the signal is, it will ultimately cause the cell to differentiate into something very, very special. All right, same thing again right here. That slide right here. Now what I'm showing you here is all the signals. So every single time there are signals causing it to become more and more and more special. So the question is, if we know what the signal is that will cause a stem cell.
Any sophomore class officers, please report to Mr. Hatlin's room. Any sophomore class officers that are still in the building, please report to Mr. Hatlin's room.
So the power of this whole thing is, if we know what those signals are, look what we can do. This process takes place, but now this is where we come in. What we can actually do is we can isolate those cells, and then if we know what the signals are, we can make those cells go down any line that we want. What we can basically do is we can create pancreas cells, we can create nerve cells, we can create muscle cells, and that's the power of these whole stem cells. But the tricky part is what? What do we not know? We do not know what the signals are. If you think about those that have a little bit more biology knowledge, the cell is very, very complicated on the outside. Many, many structures are interacting with other cells. And every single one of these structures has a function. And every single one of these structures will receive one type of a signal. And if we know those multitudes of signals, which is super complicated to get there, we can then allow the cell to go into any kind of cell. And then what do we do with those? What about if we have a dish of nerve cells? What could we possibly do?
Trains.
Have somebody walk.
Yes.
Spinal cord.
Mr. Dobson, what could we do with some pancreatic cells?
You could give people the ability to make insulin.
Very good. All right, so the power is immense. All right, so, and we call this whole thing cell-based therapy and regenerative medicine. Regenerative, we can redo damaged tissue. So, power of stem cells, here's kind of like a slide that shows you all the things that you can do with the stem cells. The part that I just showed you is cells for transplantation, or we can do a lot of stuff. We can also do just basic research with it to find out more about how these cells grow, why they differentiate, how they do this. This is the part down here, biotechnology. Instead of using pigs or animals to do drug testing, we can now use only cells for drug testing. So a lot cheaper. You don't have to go through expensive trials and so forth. So the power of these things is immense if we can actually figure this out. But again, what we need to know is how do we get from here to all these different cells. All right, so what kind of stem cells are there? There's 3 types. First of all is the adult, and we also call it the somatic stem cell. And these cells exist throughout our body. Every one of us has a whole bunch of these guys. And those types of cells are, for example, cells that give rise to blood kind of tissue, cells that give rise to nervous tissue, and so forth. Their job basically is to repair any kind of damaged tissue and cells that we already have, and to repair what's been lost through wear and tear. The other ones that are existing are embryonic stem cells, and the HESCS stands for human embryonic stem cells. And these guys give rise to the entire organism. So those are the ones that we have. Remember the hollow ball that we started out with? Those are the very, very early kind of cells. And then the last ones, those are cells that we do not have in our body. Those are called induced pluripotent stem cells. You guys all look in your sheet, what's pluripotent for? Is that the best way, the least committed, or is that already a little bit committed? Already a little bit committed, one step committed. Okay, so it's basically the same line as we wear as that hollow ball, the gastrula, when it starts to form those 3 layers. All right, so these are 3 types of stem cells that are there. That went a little too fast. Okay, question is, which one of those 3 will be best for research or for regenerative therapy, and why? Why embryonic?
Because it's the most undifferentiated.
Yes, those are the cleanest slates possible. So we can do anything with them that we want. However, again, we need to know what are the signals that make them go down one way or another one. So, any issues with those? To get them is a little bit on the tricky side. Which ones are just as potent but not as maybe tricky to get, maybe not as morally controversial or ethically controversial? These guys down here, what these things are, these are regular body cells, and somebody figured out, now if you remember the regular body cells, they're already a little bit more committed. What was the next step of commitment? It was totipotent, pluripotent, and then we get to multipotent, okay? So these are basically kind of like multipotent cell. They're already quite committed, but somebody figured out how to get them back to this stage right here. So they're reversing the signal. So basically they said, I can make this go into anything I want to. So basically, what are these guys very similar to? Absolutely, they're very similar to this one right here. Plus, there's a heck of a lot less controversy about getting these things. So, how do you get the human embryonic stem cells? Where you get them from is people that went through in vitro fertilization, basically that received or conceived a child outside of the womb. Typically, what they do is they fertilize a whole bunch of eggs, not just one. And they only then they let them go for like up to stage, I don't know, 6 or 7 days when we have this like close to the ball kind of scenario, and then put them in the freezer. And they only implant a couple of them because you don't want to have octomom, 8 babies growing. You want to have a couple less in there. So the rest is stuck in the freezer, and typically they get destroyed. Or you can decide to donate them to research. For those that got donated to the research, so you use your hollow ball, what you basically are doing is you kind of like separating the cells, and you can see the microsurgery that you have to perform here because you don't want to damage those cells. And you put them basically on a petri dish. And the petri dish has mouse feeder cells in there. So those are mouse cells, and the reason why you have to put them on there is because, well, what do cells typically do? Do they just float around? I guess cells aren't floating around, or are they attached to something? They're all attached to something. So they need to have a layer of cells that they want to be attached to. That's one reason why they're there, plus they provide nutrients for them. And then they let them go through a couple stages, and after certain criteria that they pass, they can basically then say, yes, I have an embryonic stem cell line. And those embryonic stem cell lines, they can then do research on trying to figure out how to make it go down the set path that I want to by giving it the set of signals. All right, so now that you have a dish of stem cells, how do you stimulate them to differentiate any kind of path? So the idea is you want to give them the same environment that they would see inside the body, which means that you need to know what is the environment. So there's a lot of research going on. What exactly is it that the cells are normally used to? So there's 2 things you can do, a couple of things. You can change the chemical composition of the culture media. Culture media is the liquid that you provide them, the food, the nutrients, the buffer to make sure that it stays pH stable and so forth. And you can also change the surface of the dish, because you remember human cells or animal cells like to stick to something, they don't float around. And then something also is that is not at all what normally happens. You can also insert genes, and through the gene insertion, you can also allow them to commit down one path or another one. So there's been lots and lots of trial and error. This whole thing is, I think maybe 15, 20 years in superpower. Very, very fast kind of research, but lots and lots of money has been spent so forth. And there's only a couple of cell lines that are actually established up to this point. All right, so very close to home, UW-Madison, this guy right here, and I always tell the AP kids, I promise you guys he's going to get the Nobel Prize at some point, maybe even the time that you guys are going to be there. He was the first guy that ever was able to have the embryonic stem cells. And what he also was, he was the very first guy, and that was only 6 years ago, that he made those induced pluripotent stem cells. Remember, those induced cells are the ones where you're taking cells from your body and then you make them backwards. You reprogram them backwards. Basically, you are scratching off the signals so that they can become anything that you want to. So this is just a half hour down the road from here. All right.
I have a question.
Yes.
No one else in the world has done it?
Oh, yeah, they have been others,
Yeah,
but he was
yeah.
the very first one.
He
Absolutely.
was. Okay.
And he's actually he's the leader in this whole field.
Okay.
So he gives a lot of, for those that are close, he gives a lot of good talks. He's a very comfortable individual to be around. He's not at all stocky like some of the researchers are. He's a really good guy.
Because in other countries they are using.
Oh, absolutely, yes. So where are things at right now? With embryonic stem cells, as I said before, it's not very old, so we still are totally in the infancy of the research. There are a couple of clinical trials right now that are trying to see whether retinal cells can be like reprogrammed so that your like is it macular degeneration when your retina kind of detaches and you cannot see very well, to kind of reverse that part. So there's not much going on. Induced pluripotent stem cells, there's some trials going on. I think this one is just like 2013, so a couple months only, where they started clinical trials. And clinical trials are typically on the order of like 10, 15 people, if even that many. And with adult stem cells, this has been going on for a long, long time. Have you ever heard of like a bone marrow transplant? Whenever you do that, you're giving somebody like a cell line that's already partially committed to become blood tissue, bone tissue, and so forth. So that's been going on for a long, long time already, a couple of like decades already. All right, so far out there, where are we at? This thing right here is a little bit hard to tell, and I didn't know what it was until I actually saw the little thing in the bottom. This is a heart, and what they've done with this heart is they've taken out all the cells. So basically all you have is what's left over, like the heart tissue, the connective tissue, and all the cells have been dissolved away. And the goal is to kind of reseed this with stem cells and now allow those stem cells, if you give them the right signals, to regenerate a heart, so basically to repopulate and become all heart cells. That's one thing. This is totally research down the road now. It's not at all being used. This, on the other hand, is an artificial windpipe, and the artificial windpipe, again, is a scaffold that has been seeded with the individual's stem cell from his kneecap. And they basically regrew the windpipe, and the reason why he lost the windpipe is because he had cancer in the throat, so it's a pretty nasty thing. And they replaced the windpipe and basically has a brand new windpipe. So these are the things that are going on right now, and I think, yes, there's a video out there. It's about 4 minutes long. I don't know if anybody wants to look at this. Anybody interested in this one? Can I do this? How do I do the hyperlink on here?
Just click on the blue stuff, and if it's set up right, it should. It may take a minute to pull it up, but it should pull it.
The volume?
I believe it's still on. Luke, did we turn that off after the music?
I think we're good.
Okay.
So what this video is about is how to get a flat stomach. There we go.
Honestly, this was all shot with my new Nokia Lumia icon.
Wait, those are the stuff?
Not really. I don't have much feeling on the top of the leg anymore because of the nerve damage. So, but like on the inside, it's real sensitive, and here back, it's sensitive.
This is Henry Fountain of the New York Times. Sergeant Ron Strang was injured by an IED on a patrol in Afghanistan in 2010. He lost much of his left thigh.
And I just remember looking down and seeing my leg was just kind of splayed open. I could see all the way down to the bone.
After 12 surgeries, Sergeant Strang was chosen for an experimental therapy that sounds a bit like something out of a science fiction movie. The plan was to try to regrow some of his thigh muscle using a material called extracellular matrix, or ECM.
Within that first week of me talking to him, I had the paperwork signed out, had the surgery date, physical fitness plan.
ECM consists mostly of proteins. It helps keep cells in their proper place in an organ or tissue, but it does a whole lot more. Dr. Stephen Badylak has been studying ECM for 25 years.
Extracellular matrix is the glue that holds all of the cells of our body together. Extracellular, outside of the cell material, is loaded with information that tells cells what to do. The extracellular matrix can be harvested from any tissue by taking that tissue and then getting rid of all of the cells, and so that you're left with only the matrix. Our extracellular matrix is very similar to a pig's extracellular matrix, or a rat's, or a dinosaur's, even a tree for that matter.
Sergeant Strang received ECM that had come from a pig urinary bladder.
This is the outside of the bladder, and what we're going to do then is remove all this that you basically see here is the muscle fibers. The next step is to just try to loosen up the muscle fibers. And what I'm going to do is take the scissors and make a shallow incision across the midline, through the muscle layer down to what is the matrix. We're then going to start peeling those muscle fibers off sort of by hand in a sheet. So I'm going to grab that muscle, I'm going to start at the middle and work my way over, and then kind of keep coming back and working further and farther down until I've stripped off all that muscle in a single sheet. We'll then flip it over and do the other side. So what you're seeing here is the extracellular matrix along with some muscle fibers that are still attached. At that stage, it's now prepared for various chemical baths that will help remove any DNA, any cells that are left behind. At the end of that stage, it's decellularized. So this is actually this bladder processed and dried. It's kind of a parchment paper like construct. And then what we do is we cut them into nice uniform shapes and sizes for convenience sake.
When they put the ECM in, it was just a small strip on this side that laid in.
A day after the ECM was sewn into Sergeant Strang's thigh, he was exercising the leg. This is a critical part of the treatment, doctors say.
One of the biggest differences about this therapy versus present-day standard of care is that we put them into physical therapy immediately, literally the day after surgery or 2 days after surgery, because one of the mechanisms by which we believe this approach works is that as the biologic scaffold begins to get degraded and broken down in the body, which is starts to happen immediately, it releases signals that recruit the body's own stem cells to the site. Those stem cells get there, and then they say, okay, now what do you want me to do?
The study is funded by the Department of Defense, and most of the patients so far are wounded veterans. Dr. Badylak says his goal is that this will become a standard treatment for some people who've suffered significant muscle loss. 8 months after the surgery, Sergeant Strang, who could barely walk on his left leg, is now running and biking.
They pretty much gave me a large part of my life back that I wasn't able to do before, and I was almost to the point of just being used to it being that way, because I didn't know, that this was even possible, that they had this out there, that they were able to put a pig's bladder into my leg and turn it into muscle.
There you go, the power of stem cells. So you just need something for those stem cells to hold on to. You need to know what the signals are. How did this guy get the signals? His own body gave him his own signals. So all we need there is a scaffold for the cells to hold on to. Signals were provided, and you can read blood tissues. That's kind of where things are going, and this has already been done. So, all right, any questions? Mr. Rump is ready to go.